Hi, everyone. I'm Marcus. I'm a second year PhD student at UCL, and my talk is titled Tripping the Switch, the Role of Psychedelics in Rewriting Addictive Memories. So I imagine there's a bit of variation in our prior knowledge of these drugs, so I'll quickly go through the basics about them. The term psychedelic is derived from Greek. So psyche means soul or mind, and delos means to manifest. So psychedelic means mind manifesting. Regarding how they fit into drugs, this is a good way to imagine it. We have psychoactive drugs, which include all sorts like alcohol, nicotine, SSRIs, you name it. And then within psychoactive drugs, we have hallucinogens. And importantly, this is not synonymous with psychedelic. There are some types of drugs that are hallucinogens, but not psychedelic. So, for example, dissociatives, which include ketamine or nitrous oxide, and deliriants like nutmeg in high enough amounts. And then we have psychedelics. And what exactly is classed as a psychedelic is still debated. But when I say psychedelic, I'm referring to classical psychedelics, which include psilocybin, LSD, mescaline, and DMT. What do they do to people? Well, quite a lot. So in high enough doses, they drastically alter someone's conscious experience. And this can include changes in perception. So things may seem more colorful, or hallucinations might occur. Objects around you may appear to move or morph into different things. You might imagine objects or beings or sounds around you that aren't actually there. There might be some quite strong emotional effects. So they can induce feelings of joy, euphoria, or love, or perhaps terror or fear or anxiety. It really does depend. And there are some physiological effects as well. So perhaps it depends on the drug again, but perhaps increased heart rate, pupil dilation, and impaired coordination. What's crucial to bear in mind is that the effects of these drugs are highly context dependent. So the mindset you are in and the physical environment you are in massively predict the effects that you experience. Now, as well as being used for general exploration of the mind and other recreational purposes, psychedelics are being used more and more in a medical context. And they seem, from early results, they seem to show quite promising transdiagnostic clinical efficacy. So in other words, they're showing promise in treating multiple psychiatric disorders like addiction, PTSD, OCD, depression, anxiety, lots of different things. The drug that my research group is interested in is DMT in the bottom right here. So that is a short-acting psychedelic. And while psilocybin and LSD can last for 6 to 8 hours, DMT lasts just about 10 to 20 minutes. It's a short-acting, powerful, cognitively disruptive drug. Hopefully you can appreciate why these drugs are of interest to us. They're quite a unique class of psychoactive drug. So what are we doing with them? Well, that's where the Unity project comes in. So Unity stands for Understanding the Neuroplasticity Induced by Tryptamines. And that's exactly what we're trying to do. So tryptamines are a class of psychedelic, and they include DMT. And we're trying to understand the plasticity-related effects of these drugs. This is our big psychedelic project at UCL. Importantly, this is a mechanistic trial. It's not a clinical trial. It's a mechanistic study. We're trying to understand the drug because a lot of the research in this space at the moment is clinical. We're taking a step back from that and looking at how this drug works, what it does to the mind and brain, trying to get a holistic understanding of DMT. It's an absolute octopus of a project with various arms and components and things, so I won't go into all of it today. I'll be focusing on the part which I'm involved in, which is this. What are the effects of DMT on memory, and can we harness these effects clinically? So a very brief high-level background on our understanding of memory before we continue. We start off with encoding. So when you're exposed to new information, new short-term memories are encoded. And then some of these short-term memories are consolidated into stored memories, otherwise known as long-term memories. Now, until about 30 or 40 years ago, it was thought that once a memory has been consolidated, that's it. It can't be changed. It can be covered up by new memories, but it can't be fundamentally changed itself. But a bunch of research came out in the 90s basically challenging this notion, showing that these long-term memories can be reactivated or retrieved, and then they enter this unstable labile state. And when they're in this state, they are vulnerable to change. So they can be changed when they're in this state, and then they get reconsolidated back into long-term memories and become stable again. So this process of reconsolidation is key for what I'll be talking about today. So why is that relevant? Well, in short, the logic is this. So addiction, trauma-related disorders, and phobias can be conceptualized as disorders of memory. They're not inherent. People are not born with them. They may have particular genetic predispositions to them, but they are not born with them. They're acquired when our associative learning processes malfunction and we end up placing increased emphasis or weighting on fear-related or reward-related memories. Ultimately, by definition, these disorders are learned behaviors, and by extension, if they can be learned, they can be unlearned under the right conditions. The learned associations that underlie addiction in particular are referred to as maladaptive motivational memories, or MMs, as my supervisor calls them. These are memories that link environmental cues to a drug's rewarding effect. So, for example, the smell and sight of a pint being associated with alcohol's rewarding effects. Let's go back to this figure and think about how this applies to alcohol and our connection to alcohol. So imagine you enter a pub for the first time. You've heard a lot about beer, but you've never tried beer yourself. And you think, oh, I'd like to try beer. I'd like to see what all the hype is about. So you get a beer, and it's your first beer. So you feel the effects after just one, and you think, wow, that's a nice little buzz I've got after that one pint. Wow, this pint makes me feel good. You've created an association between the taste and smell of beer and alcohol's rewarding effects. This association that you made is stored in your long-term memories. So you think, oh, that pint I had a few days ago was great. And then over time, you're exposed to more pints. You go to the pub again and again. You get pints again and again. Oh, look, another pint, lovely. Every time you are exposed to a pint, that association that you have is reactivated. The more you do this, the stronger that association becomes. And before you know it, you find that when you walk into a pub, and you get exposed to all the typical pub cues, like the smell of beer, the dark brown furniture, the sticky tables. Maybe that's more of a UK thing, I don't know. You get exposed to all those cues, and then you think, wow, I really fancy a pint. This association ends up becoming quite powerful because it's repeatedly reinforced. Now, in most people, this is perfectly normal, it's perfectly healthy. People drink at a healthy level, but it can become pathological. Like I said on the last slide, you can get these maladaptive motivational memories. So memories that encourage you to drink to an unhealthy extent. To demonstrate how powerful this association can become, I'd like to show you this. So researchers have tried to identify what this association looks like in the brain. And to do that, they get a bunch of alcoholics into a brain scanner and they show them various pictures. Some of these pictures are of alcoholic drinks, some of them are of non-alcoholic drinks. And then what they do is they compare the brain activity resulting from these two image types. So they can identify what parts of the brain are selective to images of alcohol in alcoholics. And this is what they find. So this is the alcohol selective brain activity in alcohol dependent patients. And as you can see, it spans so much of the cortex, and it shows how powerful and controlling this association can become. And it leads to a very strong urge to drink. You don't see activity like this in healthy drinkers. This is what those MMMs look like in the brain. That's just one example of how a memory or an association can change over time. We all know that memories change over time. They often do throughout life. The brain is constantly altering stored memories so it can better make predictions about the world and maximize reward and minimize surprise and error. The question is this something we can harness clinically? So can we reactivate the memory, put it into this labile changeable state, interfere with the storage procedure, and then therefore weaken the memory that gets reconsolidated, making someone less controlled by these powerful maladaptive associations? So back to this figure again, this bit here, this modification aspect is what we're interested in. Can we modify this process of reconsolidation and interfere with it? That is what my work focuses on. That's pretty crazy, pretty wild, pretty clockwork orange. Does it actually work? Well, yeah, it seems to. So over the past 20 years, lots of studies have been done on this. And the evidence shows that we can reactivate a memory, destabilize it, change how it's stored, and weaken it so people become less vulnerable to their phobia or less inclined to drink that pint or take that drug. So this is a meta-analysis I've been working on. So it's a big collection of studies. Each of these purple blobs is a study. And if they are to the left in the midline, that means they showed that memory reconsolidation interventions worked. If they're to the right, it shows that they didn't work. And you can see that on average, the vast majority of studies are to the left in the midline. And at the very bottom here, we have the average effect size of these studies, which show that, yes, there does seem to be promise with this type of intervention. So I've told you about psychedelics, I've told you about alcohol-related memories, and I've told you about memory reconsolidation. Now it's time to bring it all together. The question we're asking is this: can psychedelics be used in the process of memory reconsolidation? And does this actually explain their already observed therapeutic effects in the context of memory-based disorders? This idea has been proposed a few times now, so it's about time that it was actually tested. Why do we think that psychedelics might have potential here? We have a few reasons. So firstly, psychedelics appear to have plasticity-inducing properties. So in cell samples and the zebrafish embryos, it has been shown that psychedelics facilitate change on a cellular level. Secondly, brain scanning studies have shown that psychedelics bring about huge network-level disruption across the brain. And this sort of global disruption of typical functioning has been shown to help other memory reconsolidation interference interventions. And finally, there is some EEG evidence. So EEG is when you put a cap on someone's head and it measures brain waves. Psychedelics appear to disrupt alpha brainwaves and theta brainwaves. And these are two types of brainwaves that have been indicated in memory. So if we can interfere with these types of brainwaves, perhaps we can interfere with memory. So the rationale is there. And interestingly, it has already worked. It's worked in 2 rodent studies now. So the great thing about this field of research is that memory is so fundamental in the animal kingdom. It's evolutionarily conserved for so many species, from crabs to snails to rats to humans. So rodent studies are a great starting point to test whether psychedelics have potential in this context. Ayahuasca, which is a DMT-containing psychedelic brew, has been shown to interfere with fear memories in mice, and psilocybin has been shown to interfere with alcohol-related memories in mice, which is quite promising for our purposes, and I'll explain why later. In the psilocybin study, they tested a few different bits, including whether psilocybin by itself had an effect on drinking, and they found that it didn't. Then they tested whether psilocybin given after alcohol memory reactivation had an effect, and it did. We can see in the graph here, the blue bars are mice in the psilocybin condition, and the white bars are the mice in the placebo condition. And the larger the bar, the more alcohol-seeking behavior the mice exhibited. And so you can see there is a very clear difference in the white bars and the blue bars. So mice in the psilocybin condition displayed significantly less alcohol-seeking behavior compared to the mice who got placebo. So in short, what we're doing is testing whether this works in humans in a study way more conducive to people than mice. Obviously, it's a big jump from mice to humans, but that's exactly what we're trying to do. The Unity project is the first study to test this idea of psychedelic-assisted memory reconsolidation interference in humans. This is our big messy study protocol. I won't go through it all today, but the crucial part is this middle bit here. So section 2. Section 1 is the baseline. Section 3 is a week after. So we've got some baseline measures and post measures, and we compare them. But section 2 is the important bit. where we have AMR, which stands for alcohol memory reactivation, and that's followed by either DMT or placebo. And then there's also a no reactivation condition here, followed by DMT or placebo. So we have a factorial design, see whether this intervention works or not. And then we follow participants up for 9 months, and we use the timeline follow back, that's the PLFB, to monitor their drinking and see whether there are changes in alcohol consumption. We plan on recruiting hazardous drinkers, not alcohol-dependent individuals, because that would make this a clinical trial, and there are various reasons why we don't want that. So we're recruiting a subclinical population, and this is our memory reactivation procedure. So participants are presented with a bit of beer or a bit of cider, and they're told to pick up the drink, smell the drink, prepare to drink, and then they're told, don't drink, put the drink down. And that brings about a prediction error, which is thought to destabilize the reward association. Shortly after this, we administer them with DMT within that memory reconsolidation window. And then DMT, given that it's a short-acting, cognitively disruptive psychedelic, we're hoping that it interferes with the reconsolidation of that alcohol reward association, therefore weakening that association and making people less inclined to drink. Again, sounds pretty wild, but it has been successfully implemented before, alongside ketamine in a study run by my supervisor, Professor Ravidas. What do we expect exactly? This is our main hypothesis. So we suspect that memory reactivation alongside DMT will significantly reduce alcohol consumption compared to the other conditions being conditions without DMT or conditions without memory reactivation. It's something a little different about this intervention compared to other psychedelic-assisted interventions that you may have noticed. This is not psychedelic-assisted psychotherapy. We are targeting implicit memory systems. It's quite unique in that sense. But crucially, that's not to say that this is some kind of cold approach, some kind of heartless approach. We do provide plenty of support to participants. But we provide psychoeducation before the intervention. So we give people the tools to cut down on drinking by themselves, and we extensively educate them about what to potentially expect from the DMT experience. And we provide preparation materials so they feel ready when they go into it. And we provide support both during and after the experience too. If our intervention works, it could indicate that memory reconsolidation is a harnessable therapeutic mechanism of psychedelics, and it could pave the way for psychotherapy-less psychedelic interventions, which would make psychedelic-assisted medicine much more accessible and viable on a large-scale healthcare setting. And that's a very promising selling point of this type of intervention. Regarding what's next, I've got to leave it there, unfortunately, because we don't have any proper data yet. We've done a bunch of piloting, though, and people are responding very well to the drug. And we will be recruiting 120 people within the next year and a half. And here is a QR code to our website if you want to learn more about our work and follow along with what we do. And this is my wonderful team, who I'm very grateful for. Thank you very much.